Today I am going to talk about cancer. As you know, among various diseases, cancer actually has become a big threat to human beings globally. As per the Indian population census data, the rate of mortality due to cancer in India was very high and also alarming. About 8 lakhs and 6,000 existing cases of cancer in India. It may be that more than that during the last century. Cancer is actually the second most common disease in India, responsible for mortality with about 0.3 million deaths per year. So cancer actually is really very important disease, needs for further intervention, and there is a scope of exploring cancer biology in basic science and also in the level of translational research. So what I am going to do, that is I just am planning to produce some lectures of cancer biology which will cover basic facts of cancer, and depending on this, later on we also will start to produce some lectures which will cover the translational area of cancer biology. Cancer actually is a group of disease characterized by uncontrolled cell division or uncontrolled cell growth. Cancer cells are not foreign, they are actually derived from the normal counterpart of our body, or we can say tumors or cancers are growths that derived from normal tissues of our own body. Basically, the 19th century discovery that speaks that all cells of an organism derived from fertilized egg, that lead to the realization that tumors are growths that derived from the normal counterpart of our body or from the normal tissues of our body. So tumor mainly is of two types, as you know from our school books, school biology books, benign and malignant. As you know, benign tumors actually are non-invasive or less harmful in contrast with the malignant tumor cells, which are invasive. It means that malignant cells have the capability to form secondary tumors. It means that from the primary site it can move through a process is called metastasis and can give rise of its own kind on a different tissue or on a different organ of that organism, is called malignant tumors. Basically, malignant tumors are very dangerous and contribute to the 90% cancer related mortality. So basically what I am going to say, that is I am to discuss about the malignant tumor cells, how basically our normal cell of our body become cancerous, why our normal cells try to harm ourselves. This actually is very interesting. Before going to that topic, I want to say about the classification of normal cancers. Cancer actually is divided into 4 types. First one is epithelial. The epithelial origin, the cancers which are originated from the epithelial cells are called carcinomas. And next comes the mesenchymal origin. The cancers which originate from the mesenchymal cells of our body are called sarcomas. The cells of our hematopoietic system. Means hematopoietic system means the system where blood cells and lymphocytes are produced. It's called hematopoietic systems. If those cells lost the control on their division, and if they become cancerous and they divide uncontrollably, it's called the cancer of hematopoietic system, and this is called the hematopoietic cancers or blood cancers. Last one is that neuroendodermal origin. Neuroendodermal origin means those cancers which actually are originated from the cells related with the nervous system of our body. It may be the medulloblastoma, glioblastoma, and different kind of tumors which are associated with our nervous system. Virtually all cells of our body can give rise to cancer, but the most common are epithelial origin. As I told, it is called carcinoma. Carcinoma is also subdivided into two types. First one is that adenocarcinoma, second one is that squamous cell carcinoma. In adenocarcinoma, what happens? Basically, adeno means the gland, and those cancers or tumors which arises from the gland of our body are called adenocarcinoma. Basically, the glands are formed by the epithelial cells, and if the epithelial cells which are forming the gland are mutated and give rise to cancer, is called adenocarcinoma. Next is squamous cell carcinoma. Squamous cell carcinoma actually arises from the protective cell layer of our body. Means it may be that those cells who are covering our internal organs like lungs, our gastrointestinal tract, it may be the cells which are lining on the skin are called the protective cell layers. So what we have learned, that is, cancer is actually the group of disease characterized by uncontrolled cell growth. Basically, the cells of our body divide not by themselves. I mean, the decision of cell division is not governed by themselves. It is governed by the other cells of the body. Cells divide not for themselves. Cells divide for the benefit of that organism. So, cell division is tightly regulated. And when the regulation of the cell division is lost, cells become mutated and cells divide uncontrollably to give rise the disease called cancer. During normal tissue homeostasis, what happens, that the rate of cell division and cell death actually are equal during normal context. But in cancer, what happens, that cell division and growth of the cell increases. Apparently, the rate of cell death is decreased. The programmed cell death is called apoptosis. It has been seen that in cancer cells, what happens, that cancer cells actually acquire some capabilities or some mechanism by which they resist the cell death procedures. So what happens, gradually in a limited time period, cell division increases and cell death decreases. As a result, there are abnormal growth in the tissues are seen. Hallmarks of cancer. Basically, our understanding about the cancer, what we understood till the date about the cancer, that is, cancer cells actually sustain the proliferative signaling. Okay, they have the capability of sustaining the proliferative signaling processes to promote their growth. Second is that evading the growth suppression. Cell have their capability to suppress the growth of itself, but what happens when the cell become mutated and when it become cancerous, it evade the growth suppression capability of the normal cell. Third is that cancer cell actually have the capability to resist cell death. Fourth is that they enable their replicative immortality. It means that the cell has the capability to replicate itself enormously. Then angiogenesis comes. Angiogenesis is the process by which new microvascular system are originated on the tumor. Angiogenesis is very important because when the cell grows uncontrollably and makes a mass type of growth, it needs more and more nutrient and oxygen also. That's why tumor cells have the capability to express some certain kind of molecules, for say here vascular endothelial growth factors. This kind of protein molecule have the capability to induce angiogenesis, means the formation of new blood vessels. So what happens, the tumor cells produce this vascular endothelial growth factor which acts on the blood vessels, nearest blood vessel of the tumor, and induce the division of endothelial cells which lines the blood vessels and produce new blood vessel for their more and more required level of nutrient and oxygen also. Next comes metastasis. Metastasis actually is the process by which cancer cell move from the primary site to the secondary one, and metastasis is really very important in cancer biology because this is the main cause of cancer related mortality. We can say the 90% cancer related death is actually caused by metastasis. Normal tissues carefully actually control the production and release of growth factors, but cancer cells actually deregulate these signals and become the master of their own destiny. Basically, a cancer cell actually needs some different type of growth factors and to divide more rapidly to produce the extra growth or cell mass on the organ. So what happens that on the normal cell actually they have careful control on the production and release of the growth factors which actually leads to the cell growth and cell division. But in cancer cells what happens that to produce enormously they should require the growth factor in larger amount, more and more growth factors is required. That's why what they do, they activate some certain kind of autocrine signaling also, and also they influence the other cells of the tissue to produce some different type of growth factors for the proliferation of the cancer cells. This actually is very crucial for the development of cancer and tumor progression. So now I would like to describe some signaling pathways by which cancer cells actually acquire their ability to divide enormously. What happens when a cell actually divides through mitosis? After division, it has the choice to be on the G1. Then if there is required amount of growth factors, it goes to the S phase where the DNA replication actually takes place. Then it proceeds to G2. After it, it goes to the M phase. What happens for this cycle? The cell actually need the growth factor which will say the cell is this is the time that you have to divide, and cell divide according to the signal of the growth factor which is controlled by different cells. In our body we have basically some cells which are dividing first, and some cells they are actually not dividing. So those cells who are not dividing, normally we can say about nerve cells which are situated on our brain, actually they are not going to divide, and we can say that they are in the quiescence stage and it is irreversible. Also we can say the liver cells of our body, they are also in the quiescence stage, but it is reversible. It means that when there is any need, liver cell also can divide and can give rise to different daughter cells. But in nerve cell what happens that they have not the capability to divide, and after division during the normal development, it is derived from the neuroblast. After division what happens that it remains same for the lifetime of that organism. But on the epithelial cell which divides rapidly, for say the lining of the gut, the skin cells, these epithelial cells actually divide rapidly. And what happens when the cell have to divide, it requires some growth factor which actually regulate the division of the cell. Basically growth factors are certain kind of proteins which are secreted by different cell, and it comes to bind with the specific receptors which are present basically on the membrane of that particular cell, and after binding it exerts some different kind of cytoplasmic signal cascade, we can say the downstream signaling cascade, by which the cell come to know that yes, this is the time that you have to divide. In normal cells this takes place, but in cancer cells what happens? Cancer cells become actually the master of their own destiny. What they do, they start to express the genes which are producing the growth factors. Okay, so what happens, actually we know that all the genes of organism have the same genetic makeup, but different genes are expressed in different cells. As we know, it's called the differential gene regulation. What happens in cancer cells, they actually are not they actually start to express those genes which are required, or which are, we can say, which are crucial for the development of cancer, by expressing those genes which are producing this kind of growth factors. We can say through autocrine signaling. Autocrine means the cell is producing its own growth factors which actually should not be expressed during normal counterpart of the human body. So cell actually needed some different kind of growth factors to move from the G1 to S, and there is called a point, it's called the restriction point, where actually cell make their decision, go and no go decision. Okay, so right now I am going to discuss about the different growth factors which are required for the division of the cell. So basically growth factors actually are required for the normal division of the cell. What happens, some scientists actually took some cells from tissues, and they have allowed those tissues to grow on petri dish. Petri dish actually is culture media where all the nutrient which are required for the normal growth of the cell on the human body are created here, and all the nutrient are put into the petri dish, and when they put the cells here, cells start to divide, but after some certain division, cells stopped their division. After, if you put the nutrient also, then also the cell does not divide. What happens? Some scientists actually added serum onto the medium, and cells resume their growth and divide, and again cells started to divide actually. Serum actually, how we can obtain the serum? Serum, we can say it may be the BS, I mean bovine serum. Bovine serum actually means that we can obtain it from cow or cattle. Serum, how we can make the serum? We have to take blood and allow the blood to clot, and after clotting, what we have, the yellowish color of fluid on the test tube, it's called serum. When the clotting procedure is taking place, what happens that actually in our blood we have platelets. These are enucleated cells, they don't have nucleus. When the clotting takes place, these enucleated platelet cells express, they give, they secrete some certain kind of growth factors. We can say it PTGF. PTGF actually means platelet derived growth factor. Basically, it's the protein, and there are specific receptors present on the plasma membrane of the cell for platelet derived growth factor. When we add serum to the culture medium containing platelet derived growth factor, these growth factors bind on the membrane of the cell, and the cell gradually start to divide. So it means that this signaling molecule, this is basically a signaling molecule or growth factor having the capability to induce cell division. For like PTGF, numerous growth factors have been identified. Among them, EGF is also important in cancer biology. It means that epidermal growth factor, then fibroblast growth factor or FGF also discovered, NGF, nerve growth factor also discovered. So these growth factors have the unique capability to induce division on the cell, and these are the protein type molecule, and there is unique specificity on the protein, we can say it ligand, and on the receptors which also are present on the membrane of the plasma membrane of the particular cell. So these molecules actually come and bind on the plasma membrane, and it exerts its specific function, which is resulting on the division of the cell through downstream signaling cascades. But what happens in tumor cells, in cancer cells, what happens? This kind of growth factors are produced by themselves, or the tumor cells or cancer cells, they influence the other cells of the tissue to produce this kind of growth factors for their division of the for their own division. So these growth factors actually plays very critical role. Later on we will discuss about the therapeutics of the cancer biology, where some certain kind of molecules have been discovered which have their structural analogy with PTGF or EGF, and this molecule have the capability to bind with the extracellular domain of the receptor which are present on the cancer cell, and they irreversibly bind with the receptor and inhibit the binding of EGF or PDGF like growth factors on the receptor. Thus, they inhibit the downstream signaling cascade and the cells start to die. And the cells lost their capability to divide. So, in next class, we will discuss about the other points of the hallmarks of cancer.